chapter number 7, that is world of microbes. Yes, world of microbes. Till in the previous class I have talked about the cells, okay, classification of cells. Yes, about the plant cell and animal cell. Yes, now let us go to the subject, that is the world of microbes. What are microbes? Yes, what are microbes? Microbes are tiny organisms present in our body or outside. Yes, these microbes can be seen only through the microscope. Okay, these microscopes we cannot be seen by the naked eye. We cannot see them directly. Yes, okay. They are very small, minute. You cannot see with your naked eyes. So what are the types of microbes here? He has given you so many types of microbes. There are 5 types of microbes, that are the bacteria, yes, virus, protozoa, the algae and the fungi. These are the 5 types of microbes. Okay, children. Yes, now some bacteria or some microbes, they are helpful to us. Okay, they are helpful to us, and some microbes are harmful to us. Okay, they are ಒಂದಿಷ್ಟು microbes ನಮಗೆ help ಮಾಡ್ತಾರೆ. How to fight against the diseases. Fight against the diseases they are helping, and some are damaging our body. Okay, children. They are giving adverse effect to our body. So let us go detailedly in this lesson. So you find so many things around us. Some are living beings and others are non-living things. Yes, you can see so many items around your surroundings. They are living things and non-living things, both. Or those living things, you find variety of plants and animals. Living things means they are plants and animals. Yet there are so many organisms which cannot be seen through our naked eye. As I told you, some organisms are there which are, they are also living things, but we cannot see them with our naked eye. These organisms are called microorganisms. So these are microorganisms and so they are microorganisms. You have read about the classification of bacteria, fungi, protozoa, algae and virus in an earlier chapter. So all these are microorganisms. Now he has given you a activity number 7.1. Let us see what is there in this activity. Collect pond water in a small clean glass bottle. He is telling you to collect a pond water in a small clean glass bottle. Yes. Place a drop of water on a clean slide. You take 1 drop of water on a clean slide. Means you are having that blood test. For doing blood test, they are taking that slide of glass now. In that slide of glass, you take a drop of water. Add a drops of methyl cellulose to slow down the movement of protozoa. So place a cover slip over the above suspension. Examine the slide under low power 10x and high power 40x objective. Do you find a tiny organism moving around? Yes, these are microorganisms. So a drop of water naga ಸ್ವಲ್ಪ ಒಂದೇ ಡ್ರಾಪ್ ಆಫ್ ಸೆಲ್ಯುಲಸ್ ಹಾಕಿದ್ಮೇಲೆ ನಿಮ್ಗೆ ಅದು ಪ್ರೋಟೋಜೋವಾ ಮೂವ್ ಆಗೋದು ಕಾಣಿಸ್ತೈತಿ. ಅದನ್ನು ಲೋ ಟೆಂಪ್ರೇಚರ್ ಹೈ ಟೆಂಪ್ರೇಚರ್ ಒಳಗೆ ಅದನ್ನು ಅದ್ರ ಮೂಮೆಂಟ್ ನೋಡು ಅಂತ ಹೇಳಿದರು. ಸೊ ಇದರಿಂದ ನಮ್ಗೆ ಏನಾಗ್ತೈತಿ, ಈ ಸಣ್ಣ ಮೂಮೆಂಟ್ ಏನು ಕಾಣಿಸುತ್ತಲ್ಲ ಆ ಮೂಮೆಂಟ್ ನಮ್ಗೆ ಸಣ್ಣ ಅದು ಮೂವ್ ಆಗ್ತದೆ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್. From the above activity, it is clear that microorganisms can be seen only through microscope. Okay, only we can see these microorganisms through the microscope only. Robert Koch. Robert Koch is a contemporary of Pasteur. Made his contribution in new procedure of staining, visualizing and growing bacteria. So this Robert Koch, he is the next Pasteur. Pasteur means Louis Pasteur ಅದಲ್ಲ, ಅವರು ನೆಕ್ಸ್ಟ್ ಇವರು ಒಂದಿಷ್ಟು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸು ಅಬ್ಸರ್ವೇಷನ್ಸು ಮಾಡ್ಯಾರ. ಮತ್ತು ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಹೆಂಗೆ ಬೆಳೆಸಬೇಕು ಬೆಳೆಸಿ ಅದನ್ನು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಿದ್ರಂದ್ರೆ. ಹಿಯರ್ he solidified liquid culture media and with arc. So he also isolated and characterized a bacilli of anthrax and tuberculosis and demonstrated their casualties role by series of experiments. Sorry children, I have taken the next page. Yes. Sorry. Now let us go to 79 page. Yes, 79 page. The pages were overlapped, so by mistake I have taken this. So now let us go to the Louis Pasteur. So who is Louis Pasteur? He is a person who founded His contribution of Louis Pasteur of France in the field of microbiology. He did a great contribution to the field of microbiology. So led to the greater understanding of human arrangements and animal diseases. So much of Pasteur's work involved growth of bacteria. Here, Louis Pasteur, what he did much work on the growth of bacteria. He developed that bacteria and he saw the what were the usefulness and the not usefulness and the harmfulness of the that of the bacteria. In the East, in the liquid culture, he developed methods of sterilizing and pasteurizing. Pasteurized 1857 observed different kinds of microbes. In 1857 he had developed so many kinds of microbes and he has associated and he has with different kinds of fragmentation, yeast cells, spirals of various size and alcohol fragmentation and rod-like lactobacteria with lactic acid fermentation. These all things he has done in the experiment. This finding led Pasteur to say that specific microbes may cause specific diseases to man. So, these different types of microbes, the special type of microbes or the specific microbes, they are causing a specific type of disease in the man. He learned in his experiment and he gave that conclusion. His crowning achievements were pasteurizing of milk, the development of technique to reduce the virulence of the infectious organisms without eliminating their capacity to produce immunity. So here in this way he developed vaccination for controlling the cholera and rabies. So he achieved he experimented the pasteurizing of milk. Okay, he found that it is helpful to us. It is giving immunity to our body. So like this he prepared a vaccine on chlorine and for cholera and for rabies. Okay. Yes, now here another one experiment is there. This experiment is at a swan neck flask experiment. You know, swan's neck is like this. Swan's neck is like this. No, like this it is there. So this type of experiment is telling you to do here. What is the experiment here? Let us see to this experiment here. Swan neck flask experiment. Pasteur had disproved spontaneous generation to sustain his theory and he therefore designed a series of swan-necked flasks. He took little broth in the flask and boiled the broth. Then he left the flask open into the air. Only the air free from dust particles entered the flask, while the dust particles and the microbes got trapped in the bent portion of the neck. After a few days, he broke the neck, took a sample of broth and examined under the microscope. He found no microorganisms. Based on this, pasteurization concluded that living organisms do not arise from non-living matter. Thus, Pasteur explained the theory of spontaneous generation. Here, what he is telling you, what he proved here, he took the experiment here, this fan like flask here. In that flask, what he did, he put a little broth in the flask. He put a little broth in the flask here. What happened? And he boiled the broth. Then he left the flask open. Boil ಮಾಡಿ flask open ಮಾಡಿದಾಗ ಆ ಒಂದು ಓಪನಿಂಗ್ ಅಂತ ಹೊರಗೆ ಹೋಗ್ಬಿಡ್ತು. ದಟ್ ಏರ್ ಥ್ರೂ ಎಲ್ಲ ಅದು ವಾಟರ್ ಬಾಯ್ಲ್ ಆಗಿದ್ದೆಲ್ಲ ಹೋಗ್ಬಿಟ್ಟಾಗ ಏನಾಯ್ತು, then he left the flask open to the air. Only the air free from the dust particles entered. So air free dust particles ಅದರೊಳಗೆ ಬಂದು ಸೇರಿಕೊಂಡವು. After few minutes he broke the neck and took the samples. ಸ್ವಲ್ಪ ದಿನ ಆದ್ಮೇಲೆ ಅದನ್ನು ಬ್ರೇಕ್ ಮಾಡಿ ಸ್ಯಾಂಪಲ್ಸ್ ತೆಗೆದಾಗ he under the microscope he found no microbe. 4-5 days ಆದ್ಮೇಲೆ ಅದನ್ನು ಬ್ರೇಕ್ ಮಾಡಿ ತೆಗೆದಾಗ ಅದ್ರಾಗ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಏನೂ ಕಾಣಿಲ್ಲ ಅಂತೇಳಿ he proved it. So Pasteur is also encouraged the brief the brief that microorganisms aware in the air. So ಇದನ್ನಂತ ಏನು ಪ್ರೂವ್ ಮಾಡ್ದವ, ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಗಾಳಿ ಒಳಗೆ ಇರ್ತಾವ, ಅವು ಹೀಟ್ ಮಾಡಿದಾಗ ಗಾಳಿ ಒಳಗೆ ಅವು ಹೊರಗೆ ಹೋಗ್ಬಿಟ್ರು ಅಂತೇಳಿ ಪ್ರೂವ್ ಮಾಡ್ದ. ಸೊ Pasteur postulated the Greek germ theory of disease, which state that microorganisms are causes of infectious disease. ಸೊ ಇದನ್ನಂತ ಅವ ಏನು ಪ್ರೂವ್ ಮಾಡ್ದ, ಯಾಕಂದ್ರೆ ಗಾಳಿ ಒಳಗೆ ಅವು ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಇರ್ತಾವ ಅಂತ ಹೇಳಿ, ಅದರಿಂದ ನಮಗೆ ಕೆಮ್ಮು ನೆಗಡಿ ಅಂತ ಒಂದು ಕಂಟೇಜಿಯಸ್ ಡಿಸೀಸ್ ಒಬ್ಬರಿಗೆ ಹರಡ್ತಾವ ಅಂತ ಹೇಳಿ ಪ್ರೂವ್ ಮಾಡ್ದ. ಸೊ ಹಿ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ಅ ಫಾದರ್ ಆಫ್ microbiology. He is called as the father of microbiology. Yeah. Next, Robert Koch. Here, just now I told you about him. So here, after identifying what he did a new procedure. He did new procedures. He like Pasteur. He also did so many experiments to taking the bacteria and growing them and taking out the yeast and cells and examining them. So he also did some experiments. After that, he proved that the disease agents can present in every affected case and absent in healthy individuals. So ಇಲ್ಲಿ ಇದರಿಂದ ಏನು ಗೊತ್ತಾಗ್ತಂದರೆ, ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಯಾರು ಸಿಕ್ಕಿರ್ತಾರೆ, ಯಾರು ಎಲ್ಲಿ ಅವರಿಗೆ ಎಫೆಕ್ಟೆಡ್ ಪಾರ್ಟ್ ಇರ್ತೈತಲ್ಲ ಅಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಲ್ಲಿ ಮಲ್ಟಿಪ್ಲೈ ಆಗ್ತಾವೆ ಅಂತೇಳಿ ಹೇಳ್ಕೊತಾರೆ. ಹೆಲ್ದಿ ಇದ್ದಲ್ಲಿ ಇರೋಂಗಿಲ್ಲ ಅಂತ ಬ್ಯಾಕ್ಟೀರಿಯಾಸು, ಎಲ್ಲಿ ಇಲ್ನೆಸ್ ಇರ್ತೈತಲ್ಲ ಅಲ್ಲಿ ಜಾಸ್ತಿ ಇರ್ತಾವೆ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಂತೇಳಿ ಪ್ರೂವ್ ಮಾಡಿದೆ. ದ ಏಜೆಂಟ್ ಕ್ಯಾನ್ ಬಿ ಐಸೋಲೇಟೆಡ್ ಆ್ಯಂಡ್ ಕಲ್ಚರ್ ಇಂಟು ವಿಟ್ರ್. ಸೊ ಏಜೆಂಟ್ ಅಲ್ಲಿ ಎಲ್ಲಿ ಅದು ಎಫೆಕ್ಟೆಡ್ ಪಾರ್ಟ್ ಇರ್ತಲ್ಲ ಅದನ್ನಷ್ಟೇ ನಾವು ಏನು ಮಾಡ್ಬೋದು, ಎಫೆಕ್ಟೆಡ್ ಪಾರ್ಟ್ನ ಅಲ್ಲೇ ನಾವು ಅದನ್ನು ಐಸೋಲೇಟ್ ಮಾಡ್ಬೋದು, ಅಂದರೆ ಅಲ್ಲೇ ಅದನ್ನು ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಅಲ್ಲೇ ಸಾಯಿಸ್ಬೋದು ಅಂಥೇಳಿ ಪ್ರೂವ್ ಮಾಡ್ದೆ. ಕಲ್ಟಿವೇಟೆಡ್ ಇನ್ ಎ ಲ್ಯಾಬೊರೇಟರಿ ಎನ್ವಿರಾನ್ಮೆಂಟ್, ಡಿಸೀಸಸ್ ಕ್ಯಾನ್ ಬಿ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ವೆನ್ ಎ ಪ್ಯೂರ್ ಕಲ್ಚರ್ ಆಫ್ pathogen is inoculated and into the suspected loss. ಸೊ ಎಲ್ಲಿ ಅದು ಸಿಕ್ ಆಗಿರ್ತೈತಿ, ಎಲ್ಲಿ ಅದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮಲ್ಟಿಪ್ಲೈ ಆಗಿರ್ತೈತಿ ಅಲ್ಲೇ ನಾವು ಇಂಜೆಕ್ಟ್ ಮಾಡಿ ಅದನ್ನು ಸಾಯಿಸ್ಬೋದು ಅಂಥೇಳಿ ಹೇಳಿದರು. ಏಜೆಂಟ್ ಮಸ್ಟ್ ಬಿ ರಿಕವರ್ಡ್ ಫ್ರಮ್ ದಿ ಇನ್ಫೆಕ್ಟೆಡ್ ಹೋಸ್ಟ್. ಸೊ ಎಲ್ಲಿ ಇನ್ಫೆಕ್ಟೆಡ್ ಪ್ಲೇಸ್ ಇರ್ತೈತಲ್ಲ ಅಲ್ಲಿಂದ ನಾವು ಅದನ್ನು ರಿಕವರಬಲ್ ಮಾಡ್ಬೋದು, ಅಂದರೆ ಅಲ್ಲಿಂದ ತೆಗೆದು ನಾವು ಅಲ್ಲಿನ ಅದನ್ನು ಆ ಪಾರ್ಟ್ನ ನಾವು ರಿಕವರ್ ಮಾಡ್ಬೋದು. he was awarded with the Nobel Prize. He was awarded with the Nobel Prize for inventing this method of treatment. So in the year of 1905, for this work on the tuberculosis, he invented a medicines for tuberculosis. So he won a Nobel Award in 1905. So, and where do the microorganisms live, we will do in the next part of the lesson. Till then, you read the lesson, children. It is a bit difficult to understand, children, but still you have to read them. Read 1 or 2 or 3 times. Okay. We will meet in the next class. Until then, bye.